Thank you for that lovely introduction. My name is Janan. I'm a first-year PhD student in Dr. Cheyenne Sharif's lab at the University of Guelph, Ontario, Canada. And today I will talk to you about characterization of Nod-like receptor protein 3, NLRP3 in chickens and its role in Marek's disease. So to give you all some background on the NLRP3 inflammasome, it is primed through actions of toll-like receptors 3 and 4. It is constituted of three main proteins, NLRP3 being the main one as the sensor, and it actually oligomerizes into this complex that you see here. We also have the ASC adapter protein, and within this complex, we have pro-caspase 1, which then gets cleaved into its mature form, caspase 1, and that then goes on to cleave pro-interleukin 1 beta and pro-interleukin 18 into their own mature forms. And all of this ultimately forms an inflammatory and cell death cascade. We also know that dysregulation of NLRP3 leads to adverse immune outcomes and tumor pathogenesis in mammals, but not much is really known about its role in other classes of animals. And for this reason, we were really interested to look at its role in the context of Marek's disease, in which we all know it's a viral and tumorigenic condition caused by MDV infection. It leads to lymphoproliferation of CD4 cells, and it's caused a major economic burden onto the poultry industry worldwide. The good news is that we can try to improve outcomes through immunomodulation, which is great. But how has all this come together in this case? Well, what we know so far is that Marek's disease is lymphotrophic and tumorigenic, and that through immune modulations, we can try to improve its outcomes. We also know that NLRP3 inflammasome plays a major role in inflammation and controlling cell death pathways, and that its dysregulation in mammals leads to tumor pathogenesis. But what we don't know is its role in poultry and the potential role that it plays in MDV infection and tumor pathogenesis. So for this reason, we sought to characterize the presence and function of NLRP3 protein in chickens and determine its role in MDV pathogenesis. So to further look into this, we infected chickens at 5 days old with MDV and collected samples for lung, skin, and spleen at days 4, 10, and 21 post-infection. And we collected these samples, collected their RNA, and took them for real-time PCR for gene expression experiments. And we chose these time points because they're relevant for the different phases of the disease. They correspond to the early cytolytic, the latency slash immunosuppressive, and the proliferative phase of the disease. And our results will be analyzed through a two-way ANOVA and Tukey post hoc. And I said this in feature tense because we're still trying to finish the groups, but I do have some preliminary data to share with you all today. So here on the x-axis, we have the number of days after infection. On the y, we got relative expression of NLRP3 compared to our housekeeping gene, beta-actin, in the lung, spleen, and the skin. So there's two things I want to point out here. At 10 days post-infection in the spleen after MDV infection, we see a major downregulation of NLRP3, which makes sense because this is, after all, the immunosuppressive phase of the disease. And at 21 days post-infection, in the lung and the skin, we have downregulation of NLRP3 after infection. But it's a different story in the spleen, in which we have a major upregulation of NLRP3 at this time point. And again, let's just remind ourselves, this is the proliferative phase of the disease. And this is where CD4 cells are undergoing neoplastic changes. And so we can speculate that by activating NLRP3 in the spleen is trying to compensate for this neoplasm that is happening, so that through activation of NLRP3, there's also downstream activation of cell death pathways, so that the body is trying to destroy the tumors. So we do have some ongoing and future experiments planned. So currently we're trying to put a mechanism behind all this. So we're currently conducting toll-like receptor stimulation assays. We're also looking to knock out NLRP3 through short hairpin RNA constructs, and also would be interested in modulating NLRP3 in animal studies. And so to briefly wrap things up, we were able to successfully detect NLRP3 in healthy and MDV-infected chickens. And using this information, we hope to continue to understand the role of NLRP3 in Marek's disease, so that Using the knowledge that we gather, we can then target various elements in the NLRP3 inflammasome pathway in chickens so that we can try to further immunomodulate and improve disease outcomes in our poultry flocks. And with that, this is my acknowledgement slide. And if there is any time for questions, I'll be happy to answer them. Thank you.
Thank you for this for your excellent presentation, Janan. I'm opening the Q&A session, and unfortunately, I don't see any question. But we do have time for some questions, however. And I'm wondering, what are the next steps in terms of when it comes to your analysis, because you've collected multiple samples.
Absolutely. So we do want it to be sufficient number of samples so that we can actually take them for statistical analyses. This was just done kind of like last minute. I started my studies earlier this year, and with animal studies and the weight that comes with that and whatnot. It's been a little bit delayed, but it's really exciting what we saw with the samples that we collected thus far. So we're looking to first finish those groups and also concurrently work on those experiments that I mentioned to try to elucidate the mechanism behind all this.